It'd be each floor is a different class.
Okay. Well, let's go and get rolling for today. So, a couple of announcements. First of all, your grades are not going to be posted for the exam today. So, sorry. But hopefully early next week. Okay, so we'll get those grades out to you. Okay. In terms of this week, I canceled, hopefully you saw an announcement on Canvas yesterday, but I canceled the recitation sessions. We've only covered really one lecture, and it was more a review than anything else, so there's no real reason to hold recitation sessions. So this afternoon and then Sundays, and then of course this morning was canceled as well. And finally, Quiz 11, I went ahead and delayed that one. Its due date until Friday, and again, I don't like it on Friday. I know that people have a hard time remembering, but I don't know if we're going to get through the material by Wednesday to have you take it. So I don't want you to send you in unprepared either. So, okay. Friday will be the due date for that quiz. Okay. We will have, in terms of what we have left in the class, we have 2 more problem sets. We have a couple more quizzes, and then we have 1 group work assignment left, okay? And then, of course, the final exam. So we're coming down to the end. So I will be making announcements on when the group work is and when the problem sets are going to be due as we get to it. We got a little bit of time before we get to those, okay? Yeah?
So is there a problem set then that's not associated with the exam? With an exam, there's 2 more?
Well, I mean, both of them will cover, so the problem sets, the remaining problem sets will cover material that is on the 4th exam. So both of them will be associated in that sense with an exam. Yeah. So, okay. Right. Okay. Our riddle. So I can sizzle like bacon. I'm made with an egg. I have plenty of backbone but lack a good leg. I peel layers like onions but remain whole. I can be long like a flagpole yet fit in a hole. What is it? A snake. Very good. A snake. All right. So sizzle, right? Rattle. Okay. Anyway. Okay. So we did not leave off right here. It's where maybe we might get, but so let's go ahead and come back here. And this was Monday. Feels like a while ago that we left off right here. So on Monday, I introduced or reintroduced you to some of the classes of lipids. Lipid is actually a pretty general term. There's lots of molecules that would fall under the term lipid. We went through 3 classes. This is certainly not a comprehensive list that we went through, but certainly a good enough list to help us out in this class and what we're going to be talking about with lipid metabolism. The 3 that I went over would be storage lipids. These would be the triglycerides or triacylglycerol or glycerides. We talked about those. They comprise the majority of your dietary lipid intake. And then the membrane lipids, phospholipids is what we went over, although membrane lipids are pretty complex. Phospholipids is one aspect of membrane lipids. There are others, but phospholipids is what we'll kind of talk about in this class. And then finally, this last slide here that we left off with will be cholesterol and the different forms that cholesterol can take. Cholesterol, again, gets a bad rap, but it's also very important for our bodies. And in particular, where we will encounter cholesterol in this class is, of course, as the free-form cholesterol. But these cholesterol esters, which are very hydrophobic, actually, they're not amphipathic at all. And then these biosalts, which we'll talk about here in just a moment, which are definitely amphipathic, okay, having hydrophobic and hydrophilic portions. I'll mention the fact that these are precursors to steroid hormones, but we won't talk about metabolism and how those are made, okay, and pathways related to that. All right, so our challenge then for the rest of this lecture is going to be to talk about how lipids are absorbed and how they're transported through the body. A really actually fascinating topic, directly related to certainly some very important disease conditions, cardiovascular disease in particular. The major challenge that is presented to our bodies is that these very hydrophobic molecules have to be transported using the bloodstream. And the bloodstream, of course, is quite hydrophilic, aqueous, as a matter of fact, as are the internal environments of our cells. So we are going to be talking about, in particular, as we get to transport, how lipids are packaged into what we call lipoprotein particles and move throughout the bloodstream, and how we'll look at some of the biochemistry in terms of how those are processed in the tissues and also in the bloodstream. Okay, but we'll start here with absorption. How do lipids get into our bodies? Of course, this is not a very good slide. In fact, we're not going to go over this figure very much. We will talk about it kind of essentially right here, but and use this slide for that, and then we'll use some other slides for what's going on the other side of this. Okay, in terms of getting into our bodies, we eat them. Okay, so, and they're actually quite delicious. I don't know if you've noticed that about lipids, but the right combination of lipids and carbohydrates is an enjoyable experience, I think, for all of us. So, right consistency and texture. So if you haven't ate yet, I'm making you hungry and wishing that you had ate. But, go grab a pastry after this or something like that, okay? So some of you are like, that doesn't sound very healthy. Right. But it sounds good, doesn't it? I mean, okay, so anyway, so we eat these. Lingual lipase is actually an enzyme that will start to work on lipids in the breakdown of lipids in your mouth, okay? The majority, though, of lipid breakdown in terms of preparing it for absorption is going to happen in the small intestine, okay? And there are several things that we would want to mention. First of all, emulsification is really important, and that's where these biosalts, so if you come back here, this would be right, the main component of what we would consider biosalt, okay? Those biosalts, of course, they're stored in the gallbladder, they're excreted into the small intestine, and they are really very important for the emulsification of fatty acids. Problem with fatty acids or lipids as you eat them, of course, is that they can form actually a very large lipid droplets that are fairly inaccessible, certainly to absorption, and also to the enzymes that need to break them down. So the biosalt is very important in the emulsification process, essentially breaking it down into these smaller, what would be considered mixed micelles that are ready for absorption and also enzymatic activity to act upon them. Okay. I don't want to get into the biochemistry or the chemistry even of emulsification. This should be something that you covered, even in your introductory biology class. You probably talked about this, okay? I do want to talk a little bit about the two enzymes that are listed here that would be found in the small intestine. It says pancreatic lipase or pancreatic phospholipase A2. Pancreatic meaning they're excreted into the small intestine by the pancreas. Okay, they're produced in the pancreas, excreted into the small intestine. The first one, pancreatic lipase, importantly, is going to take triacylglycerides, so we'll call it TAG, triacylglycerides. Again, the majority of the lipids that we eat are triacylglycerides. This is going to break this down into monoacylglyceride. Okay, and it's going to remove then two of the fatty acids, so it will produce two free fatty acids. Okay. If you're really interested in this kind of thing, the monoacyl glyceride would be essentially the central carbon of the glycerol chain will still have its acyl chain. Okay, the two on the outside will be taken off by the pancreatic lipase. Okay. These will then get emulsified. They'll be put into what would be considered a mixed micelle, and then that is going to be absorbed, okay, into the small intestine, in the cells of the small intestine. This guy, pancreatic phospholipase, that's going to take phospholipids, so PL would be phospholipids, okay? And it's going to break off one of the chains of the phospholipid. Okay, so if you remember, come back here, phospholipids, remember, have 2 chains, and then they have a phosphate head group. You do have phospholipids in your diet, of course. And so phospholipase A will remove one of these chains. It's the outside chain again, so it'll leave the middle chain. Okay? And so that's what we would say here. We'd say PL N minus 1, if you want, plus a free fatty acid. Okay. The phospholipid, especially with one of the chains removed, can actually be a really effective detergent, and it will participate in some of the emulsification process as well. Okay. Now, after this, we're going to put a black box here. They get absorbed into the cell. Okay. Even in current literature, there's different models in terms of how these mixed micelles and free fatty acids and monoacylglycerides and phospholipids get absorbed into your tissues. There may be simple diffusion processes, meaning no mediation by transporters. There are also models that include transport proteins that are going to move these into the intracellular environment of the mucosa cells. Okay, we'll just say that it can be both. Okay, and that's probably likely because the diversity of the lipids that you're absorbing is pretty high. Okay, so there's going to be multiple means of how are these going to get into your body. Okay. Cholesterol, dietary cholesterol, is, it would seem, transported almost entirely in a mediated fashion. Okay, so I have to have a protein to do that. Okay. And it wouldn't be surprising to find out if there's really no simple diffusion of these at all. Okay. All right, so these get into your cells. Let's just put it that way. Okay, and that then ends, in some ways, what would be called this absorption from the lumen intestine. Okay. Now what we want to do is talk about then how those lipids are going to be in those small intestine cells packaged and delivered into essentially the rest of the body. Okay. So that then the rest of the body can benefit from them. Okay. And so that's what we're looking at here, reformation and packaging. Okay? And so let's talk first about reformation. The very first step in reformation is that those free fatty acids are going to be converted to what would be considered an acyl-CoA. Okay? So these long chains are going to be attached to coenzyme A group. Okay? And then those acyl-CoAs, okay, once that occurs, the acyl-CoAs, and this would take a couple of enzymatic steps. We will name these enzymes later, okay, when we come to them in other metabolic processes. I'm not going to name them now. I don't want to inundate you with a whole bunch of enzymatic names at this moment, but just give you the overall picture of this. But acyl-CoAs can be attached to glycerol and converted to then, or made into triacylglycerides. So we break those down, transport them in, build them back up. Some of you say that sounds inefficient. Too bad. That's the way it is. Okay, it's just how it happens. Okay. Now the other aspect of this would be that these can be also packaged, the acyl-CoA plus cholesterol. And those can be converted to cholesterol esters, which I'm just going to abbreviate as CE. I'm going to run into these quite a bit. Okay. Again, cholesterol esters, this is a reminder. These are cholesterol esters. Okay. Cholesterol backbone with an acyl chain attached to it. Okay. Acyl chain being anywhere between, 16 to 24 carbons long. Okay. It can be smaller. Very rarely longer than 24 carbons. Okay. Now these then, okay, triacylglycerides, as well as cholesterol esters, we're going to add to these phospholipids. We're going to add to these something called apolipoproteins. And then we're also going to add to these just straight cholesterol. And all of that put together in the small intestine is going to make what we call a chylomicron. Okay, so this is a chylomicron. Okay, and this is our first of 4 different, really 5 different lipoprotein particles that we are going to encounter. Okay, and this is how lipids are transported throughout the body, especially when they get into the bloodstream, is through these lipoprotein particles. Now, there's a couple of things that we'll want to go ahead and describe as we look at these, okay, in terms of their makeup. The first thing, generally speaking, okay, would be the apolipoproteins. Let's go ahead and highlight these. Apolipoproteins come in a variety of forms. All right, there are these classifications. So they come in class A, B, C, and E. You say, where's D? I don't know. Okay. It's in, not us, apparently. I don't know why they went A, B, C, E. Maybe, you know, the person that discovered these got a D in one of their classes or something like that, and they just didn't like D. Okay, I don't know what that is. So, okay, anyway, that was lame. But anyway, I am lame. I mean, if you don't know that by now, I'm just not going to go any further, okay? So anyway, A, these come in subclassifications 1, 2, and this is not an exhaustive list, 3, 4, okay? And I'll have to try and remember to post a paper on Canvas for you guys that kind of goes over this stuff. It's better than our book. Our book doesn't do a great job on this, but the paper that I'm thinking about does a pretty good job, okay? But A, subclassifications 1, 2, 3, and 4. B comes in 2 different classifications that will make sense to us as we go along, what are called B48 or B100. Those are the 2 that we're going to really encounter in this class. Okay. C, 2, 3, and 4. And they do use Roman numerals, that's why I'm using that. And then finally, E, these come in E2, E3. And I believe that's it. We're not going to make major distinctions in the E class. Let me check here. Yeah. Oh no, there is an E4. I forgot. Okay. Okay. All right. All right, so apolipoproteins, again, lots of these, lots of different classifications of these, okay? When we look at the chylomicron, come over here now, the chylomicron will have these, well, it'll have multiples of these, but B48, C2, and C3 are going to be the, well, we're going to talk about the function of B48 and really C2 primarily. Okay, C3 is there as well. Okay, but those will be found in what we call chylomicrons. Okay. Now, the other aspect of chylomicrons, this is an okay depiction of this. I like how they give this cross section of this, because this does help us out a little bit. You'll notice here that in the chylomicron, there's this phospholipid layer here. It's not a bilayer, though. It's a monolayer. Okay, so this is a phospholipid monolayer. You know, I'm sure that in your other classes, you've discussed the phospholipid bilayer. Phospholipid bilayer, of course, makes up membranes of organelles and of the cell and that kind of thing. In these lipoprotein particles, they are surrounded by a monolayer of phospholipids. Now, that gives them a fairly hydrophilic surface, meaning that they can be transported in aqueous environments. Okay, the core of these, as you'll see here, is comprised of triacylglycerols and cholesterol esters. Okay, so that's essentially everything that you would see kind of here. Okay, it's kind of a tangled mess when you look at it, but everything that I just colored in pink would be this core of triacylglycerols and cholesterol. Okay, and then we have here, let's try and use a very distinctive color, that monolayer of phospholipids. In that monolayer of phospholipids are also just cholesterol, not cholesterol esters, but cholesterol. Okay, and that gives these chylomicrons mobility in their membrane. Okay, yeah.
I have 2 questions.
Okay, but you can only ask one at a time. Okay.
First one, what is like the relative size of these compared to cells?
So that's a great question. I didn't look at diameter of these, so I don't know that, but it's real small. Okay, because these will be chylomicrons, and also these other lipoprotein particles. All of them can be endocytosed, and so they'll be recognized by receptors on membranes and then pulled in. They will then be absorbed. Specifically in the liver, we'll look at this. They'll get actually fused with a lysozyme, or not lysozyme, but a lysosome at that point in time. So they are significantly smaller, probably, 1/100th of the size, if not even smaller. Yeah.
And then do different people like express this in like different amounts, and then they have like different like absorptive lipids?
Yeah, well, I mean, so first of all, what we are talking about right now would be more of a dietary, right, concern. I mean, in terms of like, do people express these at different levels and that kind of thing. But first of all, just the makeup and how much chylomicrons are produced and that kind of thing is going to depend a lot on your diet. But then when you start looking at, in particular, people that have propensities for cardiovascular diseases, there can be different levels of expression of receptors that take in lipoprotein particles from bloodstream. And we'll actually talk a little bit about that. In terms of overall metabolism, and expression levels of the lipoproteins, apolipoproteins, and that kind of thing, certainly I would expect it to be different for every individual. How different and what is the variations of that and how would that, I don't know. But I would expect, due to metabolic rates of really anyone, even the things that we've talked about, glycolytic rates and all that kind of stuff, is expected to be different for each individual. Right? This is the idea of individualized medicine, that we're starting to realize that everyone metabolizes things at different rates, okay, including the drugs that we give them to treat certain diseases, due to epigenetic factors that we still don't fully understand. And so that would be true here, I would expect as well. Yep. Other questions? No. All right. So we have been, right, we're not going to talk about is there like assembly and all that kind of stuff, but in the small intestine, this will be assembled, okay? And this is what's going to then be exiting the small intestine into our bodies, okay? And that's how dietary lipids will get into our bodies. Okay? Now, I wanted to go ahead and draw your attention to this because this is our first of many, like I said, lipoproteins. Okay? And so I've got this table up here. I'll go ahead and zoom in on that table. Lipoproteins is the name for these particles. I have called them lipoprotein particles as another term that they go by. Apolipoproteins, don't get those confused with lipoproteins. Lipoproteins would be the entire chylomicron. Okay, the apolipoproteins would be the proteins found in it. Okay. You'll notice here that there's 4 of them, and there's actually going to be 5 of them that we talk about, but there's 4 of them listed on this table. Chylomicrons is at the top of them. We actually distinguish these based off of not only the identity of the proteins found in them, which can be different, but also their density and the makeup of their core. But they are named actually, except for the chylomicron, based off of their density, grams per milliliter. Chylomicrons here, and then you've got VLDLs, LDLs, and HDLs. VLDLs stand for very low density lipoprotein. LDLs stand for low density lipoprotein, and then HDLs, high density lipoprotein. So it's all based off of their density in terms of grams per mole. So let me ask you this question, our question for the day. Okay, and I'll go back to that slide and look at this, and you guys can figure this out. But as we look at lipoprotein particles, a topic that often comes up is their density. What about the composition of each of these particles makes them less or more dense? Okay, so what do you think? Talk to your neighbor about that. Text me an answer. I'll go back to that table. You can look at the makeup and you can study that and then give me an answer. What do you think? Why is one particle more dense than the other when we start looking at its composition? Okay, so on your marks, get set, go. And let me go back to that. So you got about 1:40 and get back to that slide. Okay, so here's our slide. Okay, 1:40. Talk to your neighbor. What do you think? About 35 seconds, so think about texting that answer sooner rather than later. You have about 5 seconds left, so hopefully you submitted an answer at this point in time. Let's get to the larger screen here. Okay, let's check this out, see what you said. Okay, triglyceride concentration, saturated versus unsaturated, more dense. No. Lipid to protein ratio. The number of proteins seems to be the most consistent determining factor, but also the number of triglycerides is inversely proportional to the density. Okay. Very shape, okay, less, okay, fatty, yeah, so, sorry, I don't know. Those are the honest answers. Those are the good answers, and the honest answers. All right, let me go this. I think that this is probably the most concise way to do this, and I'm trying to click on that one. There we go. Lipid to protein ratio. That's exactly right. Okay, when we look at this, coming back to this table, let me go to a different slide so I can draw on this, okay? And I'll give you a different view of also chylomicron. Okay, but when you start to look at this, okay, chylomicrons are what would be considered the least dense of all the lipoprotein particles. Their density is less than 1 gram per milliliter. And when you look at it here, their composition is really high, by 85% triacylglycerides, and only about 2% protein, and then varying percentages of phospholipids, free cholesterol, and cholesterol esters. Now, this would be an average, right? I mean, each chylomicron would be probably different if we were to look at it, but on average, this would be true of chylomicrons. As you move down this list, right, to get into high density lipoproteins, HDLs, you get more and more protein and you get less and less triglycerides, okay? cholesterol esters highest are the highest in LDLs, okay, interestingly. Now, when you start to think about this from a chemical standpoint, okay, proteins are of course made up of this subunit, and that would be amino acids. So amino acids take on this form. R groups, R groups, there's 20 of them that would be naturally occurring in a protein, but many of these R groups can have sulfur in them, or nitrogen, or oxygen, right, as you, I'm sure, are aware of. That would be identity of atoms, and that would not mean that an R group is just a sulfur atom. Okay, I do know about proteins and the amino acids, what they are. Okay, but when you look at this, now that would be versus, like, let's say a lipid. A lipid is going to be this. Okay. In particular, free fatty acid or triacylglycerides, we have some in here, CH3. You look at the identity of the atoms in proteins versus the identity of the atoms that are found in a lipid, and you just have heavier atoms found in amino acids. Okay, and so because of that, then proteins typically have a density that's anywhere between 1.3 to 1.4 grams per milliliter. Okay, whereas lipids, these will have 0.7 to 0.8 grams per milliliter density. So the more lipids you have, the less dense the particle is going to be. Okay? And so when you start to look at just straightforward names and density, you should be thinking, okay, low density means lots of lipids, more lipids than in the others. Okay, and that would be kind of the main point of this. Okay? Now, last thing to say here in terms of chylomicron, this honestly I think is a better depiction of the chylomicron, and in particular the apolipoprotein versus the one that's in the book. Okay, what you see here, this right here, okay, that kind of turquoise color thing, that would be one of these lipoproteins. I don't know which one they are trying to illustrate there. My guess would probably be B48, considering the structure of that. But these, generally speaking, are not, you don't want to think about a globular protein that's kind of wedged its way into this. These are really more like belts, and they're long alpha helical structures that go all the way around these particles. Those alpha helices are interesting because they're amphipathic. They're quite hydrophobic on one side and quite hydrophilic on another side. Okay, and so they will then interface with both the hydrophobic environment as well as then the aqueous environment outside of the cell. We'll talk about certainly not all the functions of these lipoproteins, apolipoproteins, but we'll talk about some of the functions of them as we move forward. Okay, some of them play very important roles in this metabolism. Okay. All right, so that's a chylomicron. Questions on that? No? Awesome. Now that brings us then to essentially excretion into our body. If you remember, we talked about glucose and carbohydrates. They have to go through the cell. We talked about how they use ion gradients and all sorts of things, and then they jump right really into the bloodstream. From the bloodstream, they go right to the liver. That will be true of amino acids as well. We'll look at that in our upcoming discussion on amino acid metabolism. Lipids are different, though. Lipids are going to dump into actually the lymph system. Okay, and they will dump into this thoracic duct. Okay, so in green here would be the lymph system. And this would be directly from intestinal cells in the lymph system. That thoracic duct is where they'll end up, and they will follow all the way up here, okay, and they will dump in right here into the subclavian vein. Subclavian vein is big, and it has really high flow of blood in it. And so putting these lipoprotein particles in that, okay, they get taken to the systemic circulatory system, and they really bypass the liver. Okay. This avoids congestion, okay, within capillary beds. Okay, capillary beds can be congested by these things if there is a load of it coming into the body from the small intestine. Okay, so they get dumped into a fairly significant sized vein that has fairly significant blood flow in it. And then they're distributed and diluted throughout the body. Okay, they will get to the capillary beds, but not in a high concentration. Okay. The other aspect of this, and this is important, is that unlike carbohydrates and unlike amino acids, they bypass the liver. So they go first to the tissues, and the tissues get first grabs at all these things. They're going to actually, this whole system, as they get transported through the body, that will end in the liver rather than start at the liver. It's a little bit different than what we've talked about. Now, that then is going to then bring up, okay, this, okay, what would be considered degradation of the chylomicron. And we really enter into this, what was called exogenous transport system. Okay, and your book talks about this. This is found in literature as well. There's 3 different pathways that we consider as we start to think about lipids moving through the bloodstream. One of them is in what's called the exogenous pathway. The next one that we'll cover, certainly not today, but we will talk about it on Monday, endogenous pathway, and then finally something called reverse cholesterol transport, which we'll talk about as well. Okay? The exogenous transport pathway starts at the small intestine. Okay? With a chylomicron. Okay? So that's where it begins. It's going to end at the liver with what's called a remnant. Or a chylomicron remnant. Okay? The purpose of this is to transport dietary lipids to tissues. Okay, that would be the purpose of the exogenous pathway. It really begins at what we've already talked about, the packaging of that chylomicron. Okay, so we looked at that. We talked about the makeup of the chylomicron. That's where our exogenous pathway starts. Those get dumped into the lymphatic system as discussed. They get into the subclavian vein and then distributed to all the tissues. Okay. Now, we haven't talked about, of course, the end of it, but let's go ahead and talk about this. As they're being transported through the tissues, so we've got this whole pathway down here. We've got our chylomicron, okay, via the lymphatic system. That's going to get into capillary beds. It says myocytes, adipose, mammary glands, and really muscle. Okay, skeletal muscle, myocytes would be part of that, right? So cardiac tissue would be as well. Adipose tissue, yeah, very important. Okay, but what's going to happen there is this: as they get into those capillary beds, you're going to see this process. Let's go here. The triglycerides in these chylomicrons are going to be processed to glycerol and free fatty acids. And the enzyme that's going to do that is called lipoprotein lipase. I'll just abbreviate it LPL. I'm not even sure if that abbreviation is what's used in the literature. It's easier to write, though, than lipoprotein lipase. Okay. Now, lipoprotein lipase is activated, and in fact, ApoC2 is a cofactor of that enzyme. Okay, so lipoprotein lipase is not active until C2 binds to it. Now, C2 is on the surface of the chylomicron. Okay, so if we come back here, let's go back to our chylomicron. Right, C2 is one of these proteins here. It will be recognized by the lipase, lipoprotein lipase. And when that recognition occurs, then that lipase will become active. It will start to break down the triacylglycerides. Now, come back to our makeup here. The chylomicron is 85% triacylglycerides. It's just jammed full of these. And so that protein has a lot of work to do, okay, that enzyme. Now, as we look at this, okay, I think it's important to understand this. The lipoprotein lipase, because it's not really shown here. I mean, it's just like it goes into here, free fatty acids come out, and they're magically absorbed, and there's magic that happens. And you get this sense, even when I looked at this, you get this idea that, well, there's this lipoprotein lipase that's kind of free floating in the bloodstream and then attaches itself to, well, how does all this work? Turns out that the lipoprotein lipase is anchored to endothelial cells at capillary beds around tissues. Okay, so the tissues actually, like a muscle cell, will actually express lipoprotein lipase. They will excrete it along with several other what are considered anchoring proteins, and those will be anchored into the endothelial cells on the inside of the lumen of the capillary beds. Right? So then these chylomicrons come floating through these tissues, the capillary beds interface with these tissues, are bound by the lipoprotein lipase. That starts to break off the free fatty acids. Those then get absorbed by the tissue. Right? So these will get absorbed. And that is a protein-mediated process. Okay, there are two proteins. Transporters that are involved in this. CD36 is one of them. And then a general name is this FATP, fatty acid transport protein, not adenosine triphosphate. Okay, and you say, oh, great, this is awesome. Let me go ahead and write this down. Fatty acid transport protein, and there's it's a family of proteins, okay, that are responsible for absorbing fatty acids. Now, it goes hopefully without saying, but tissues that express more LPL will get a greater load of fatty acids because the fatty acids are going to be broken down in capillary beds around those tissues. Okay. Skeletal muscle, okay, is very important in this. Adipose tissue, obviously, okay, would be where you find this. Okay, skeletal muscle uses though fats for a lot of its energy needs. Okay. Now this then, the glycerol goes off to the liver. Okay, glycerol is quite hydrophilic and therefore soluble in the aqueous environment of the blood. Okay. Generally speaking, it will be absorbed by the liver. Okay, and let's go to our next slide here. And in the liver, or the kidney, liver primarily, okay, but in the liver there is this enzyme called glycerol kinase. This is found only in liver and kidneys. All right, but it takes glycerol, so here's glycerol, and it will phosphorylate it, making glycerol 3-phosphate. Some of you say, glycerol 3-phosphate, isn't there a shuttle named glycerol 3-phosphate shuttle? Exactly, nice job, you're very, very good, okay. That's right, I mean this is, if you remembered that, glycerol 3-phosphate is a component of that shuttle. Okay, that doesn't mean that this is the shuttle, but this is one of the components of that shuttle. Okay, glycerol kinase is going to phosphorylate this. Now, that glycerol kinase, this molecule, has several different fates. Let me draw out two of them that aren't shown here, but this can be used to make phospholipids. Very important in the liver, okay? Or it can be used to make triacylglycerides, also very important in the liver. We'll talk about the enzymes that are necessary to do that. Of course, it would need acyl chains to do this, okay? Or it can go this direction, this route, where it can get into glycolysis or even gluconeogenesis, okay? In the liver, glycerol can be converted to by glycerol 3-phosphate dehydrogenase to dihydroxyacetone phosphate, and if flux in the liver is going towards gluconeogenesis, then this will go up towards glucose. Or if flux is required to go to glycolysis, then of course dihydroxyacetone phosphate will go through that and become pyruvate. Okay, so there are a couple of fates for that. If you remember when we talked about the fasted state of someone, right, as blood sugar starts to drop, we talked a little bit about some of, fatty acids can't be converted to glucose, but glycerol can. Right? This is the pathway for it to become glucose. Okay. Okay. Now, at the end of all of that, right, so coming back here, we have taken the chylomicron and we're removing, by lipoprotein lipase, we're removing the triacylglycerides. Free fatty acids are getting absorbed by the tissues, glycerol going to the liver or the kidney. And what that ends with then is this, it's called the chylomicron remnant. Okay, all that produces this remnant. And the remnant is essentially a chylomicron depleted of triacylglycerides. Right? So this is, whoa, I zoomed way in there. There we go. Ignore that. This guy is depleted of, well, tags. There's still some there, triacylglycerides, and C2. So I'll call it ApoC2. Okay? But it has ApoE in it now. Magically. Okay? So as this whole process occurs, we've seen how triacylglycerides are removed. Turns out that there are these other lipoprotein particles that are floating through, that these actually start to exchange material between one another. Okay? C2 will start to come off what are called remnants, and ApoE will be put onto it. Okay? So if you're removing C2, remember C2 is what activates lipoprotein lipase. So once that's gone, then the chylomicron can't be attached to endothelial cells outside of tissues. Okay, so they stop then, giving up triacylglycerides to those tissues once they lose C2. ApoE, okay, this is a signal for absorption to the liver. Okay? And that's essentially what you're seeing here. Chylomicron is absorbed by the liver. ApoE, if you're curious, is it will be recognized by what are called LDL receptors, or there's actually two other receptors that will then endocytose the entire thing. Once they bind ApoE, it will lead to an endocytosis process, and the chylomicron will be pulled into the liver. Okay. Once in the liver, right, so ApoE, that's what we're talking about here. Once in the liver, it'll fuse a lysosome. The remnant fuses with a lysosome. Okay, and in the lysosome, here's what will happen. It'll break down everything. Okay, protein will be broken down to amino acids. Okay, whatever triacylglycerides are left would be broken down to free fatty acids and glycerol. Geez. Glycerol. Okay, so I can't write glycerol right there. And then finally, cholesterol esters are going to be broken down to cholesterol plus an acyl group, or a free fatty acid, I guess. Okay, and that will end then the exogenous transport pathway. Okay. Which is a great place to end our discussion today. Okay. The Rockies, by the way, some of you are wondering, 4 in a row. 4 in a row, okay? Just so you know, I'm optimistic. They play again tonight, who knows? By the end of the weekend, it could be 7 in a row. That's what I'm hoping for. What's happening?
Not much. Just took a test. That's why I was late. But I have a couple quick questions.
Yeah.